So, of course, due to the snow, there are not only change of time, but also change of personnel. So I'm not Alex Binshtok, but I will replace him as the moderator of the first session. And the first session is Circuitry, and our first speaker is one of our new recruits in ELSC, Mickey London, who will talk about noise, game, ping-pong, and one little channel in between.
Okay. So thank you, Yifat. I want to thank the organizer for inviting me to give this talk. And you have to prepare yourself for a crash landing from the international spaceship down to Earth. The title is a bit cryptic, I'm sure, but it's composed of all kinds of buzzwords that will be unfolded during the talk. So we can discuss it at the end and see if anyone actually picked it up. I also have a little bit of a dilemma because I made a talk that is based on a previous work, and then I wanted to add a little bit of data and found out that there's another talk in the talk. So I will try to go quite fast on the first part, which is on published data, and concentrate on the latest bit. So to start, I want to start with this very famous work by Mehring and Sosnovsky, very influential work. What they actually tested the question of how reliable are neurons, and they did a very simple experiment in vitro in slices. Slices were loaded with synaptic blockers, so there is almost no synaptic noise. And what they did is injecting a step current into a neuron 25 times and then aligning the traces one on top of the other. And you see that while the first spike is actually very precise, later on there's a complete jitters of the spikes, and the spikes are not very reliable anymore in terms of the timing between repeated trials. However, if you take the same mean current, average current, and then inject it as a fluctuating current, the neurons become very reliable, and they can actually precisely repeat the times of each individual spike very well. On the other hand, another famous experiment by Georgopoulos from 1982, in vivo work in a monkey primary motor cortex performing a task, a reaching movement task. You see complete opposite behavior where repeated trials, while they have some general patterns, so for example, the elevation in firing rate close to the starting of the movement, the individual trials are very, very different from each other. And this actually seems that neurons are very noisy and completely unreliable. So there is this tension between fluctuation and noise that affect the spike's precision, and this will be the topic of the first part of the talk, trying to understand how precise are spikes in the brain, and what determines the temporal precision, what the role of noise in this temporal precision. And what we try to do in that work is to estimate the fluctuations that has nothing to do with any behavioral variable. So the idea is that if you do two trials, like in the Georgopoulos task, there is some level of noise that has nothing to do with any behavior, and then the precision of the spikes is affected by this noise, and we want to try to find the minimal level of these fluctuations. And the assumption was that we think of the cortex as a network of excitatory inhibitory neurons that has some dynamics in time, and we take snapshots of these dynamics, and we assume that for a start that there is no noise in the system. So if you will take two individual trials and try to repeat them, they will behave exactly the same. If we align all these neurons in a vertical way so we can actually trace them in time, you see that we have like two repeated trials, and the trials are completely identical. And these neurons are spiking on each one of the trials just as they were in the previous trial. And then we start, and then we introduce a small perturbation to one of these neurons and ask ourselves what will happen. And I will explain the motivation for that in a second. But the idea is that if you introduce this perturbation, you make a neuron spike in a times where it didn't spike in the previous trial. This extra spike will travel down the axon, will reach some postsynaptic neuron, and if it is an excitatory synapse, will create an EPSP in the neuron. And now there are two possibilities. One option is that this EPSP will actually die out, will leave no trace for its existence after a while. The other option is that the neuron will, the EPSP will come just as the spike is building up, or the membrane potential is getting close to threshold, and will provide this extra kick that will make this neuron fire. Okay, so these are the two distinct possibilities, and it turns out that if, on average, all the neurons behave like that, all this EPSP doesn't create any additional spike, then in later snapshots of the network, you'll get exactly the same picture as you've seen before, because there was no effect on any of the neurons in terms of its spiking. However, if we have a condition where on average, more than one neuron will spike, then if you take, for example, this neuron and say this neuron had spiked in a time, so this extra spike in that neuron that we perturbed created an extra spike in that neuron, then this neuron is now in a condition which is very similar to that one. And if on average we say that in that network every extra spike creates more than one extra spike, then we are in a position where this perturbation grows, and it actually grows very fast. And after quite a short time, you are in a position where many of the neurons are spiking in places that they weren't spiking before. Okay, so what does that have to do with noise? So if we are in this deterministic regime, then of course the network, if you just look at the voltage trajectory of any of the individual neurons, on every repeated trial you get exactly the same trace. However, if you're on this, in this condition, you might have the same kind of general trajectory of the voltage, but all these extra and missing spikes between trials will introduce fluctuation to the voltage trajectory, and these fluctuations, as we said, have no connection to any behavioral or meaningful variables. They're completely random, and they're just created by the property of the network to amplify these extra spikes. So this is noise. So this is what we're trying to estimate now. And first of all, we want to know whether the cortex actually behaves in this regime or in that regime. So how do we do that? This is actually, I already gave you the answer. All we need to know is that if we have an extra EPSP, whether on average it creates an extra spike in more than one of its postsynaptic targets, or it just on average dies out. And this is an experiment that we can do. Actually, we take a neuron, we patch it, we add an extra EPSP, and we look at what the probability of an extra spike. And that's exactly what we did here. We took in vivo recordings, whole cell recordings in layer 5 pyramidal cells. We injected trains of excitatory and inhibitory currents, which has a synaptic-like, you don't see it here because it's too fast, but they have a shape of a synaptic current. And if you look at the traces during the downstate, you can see that they actually create EPSPs or IPSPs comparable to actually real synaptic input. And if you average or you take the PSTH triggered on one of these currents, which is actually very small, on the order of 25 picoamps, you can see that you have a significant elevation in the probability to spike. If you take an inhibitory current, you see reduction in the probability to spike, and taken together over many amplitudes, or not many, but several amplitudes of current, if you take the probability, you end up with an estimation from a typical EPSP in cortex. You would get a probability of about 2% to get the cell to fire, meaning that if you will repeat, if you take a neuron 100 times and add an extra input to this neuron, on 2 times out of this 100, it will spike an extra spike. And this is actually a relatively very small number, but you have to remember that this neuron is connected to a few thousands of postsynaptic targets, which means that if you take this 2% and multiply by the number of postsynaptic targets, you get around 30 spikes per spike. So it means that every extra spike that you add to the network will create in the network 20 to 30 more spikes in the postsynaptic targets. I'm not getting into the inhibitory and excitatory and missing an extra spikes here, but just to conclude that this experiment actually tells us that we are working in this regime, and this is actually a regime that was described way before by Chaim and by others as a balanced state of a cortical network. I mean, this is not a one-to-one relationship, but it's a property of these networks as well. Question?
Could I just ask, and I probably missed it, how big is the EPSP roughly?
That we measured in terms of voltage?
Mm-hmm.
It's in this case, for example, it's less than a millivolt. So this is 2 millivolts, and you see this. This is a 50 picoamps transient, and it creates less than 1 millivolts EPSP. David.
But Nicky, I mean, it seems that you're implicitly assuming the probability of release is 1. I mean,
No, actually not, because these values that I said, these values that we assumed here, is actually taken from. We took it from the literature, from paired recordings that already taking into account that you have several boutons, several synaptic contacts, and a probability of release. So this is taken into account. In any case, we were trying to be very moderated about our estimations, so it's all underestimations. For example, one of the things you might notice is that we do count also these extra input in the downstates that are not going to make any extra spike. So it's all like estimation from below. Anyway, okay, so this is about far-reaching conclusion from an experiment, a simple experiment like that, to try to say anything about the whole cortex. So we try to support it with another set of experiments where we're actually doing the real thing, patching a neuron and introducing an extra spike to the neuron and recording activity in the network with a multi-unit recording electrode at the same time from the local network. And you see here's activity in the network, and you see the activity in the neuron. And when you introduce an extra spike in the downstate, you don't get anything, of course. If you introduce in the upstate, you don't know, but if you average on many of those, you see that there is a slight elevation in the probability of neurons in the network to spike. And using some assumptions that I'm not going to get into, we get to a relatively simple, similar conclusion. So to conclude, we are concluding that we are in this regime, and these extra and missing spikes are creating trial-to-trial fluctuations that are purely noise. But what is the size of these fluctuations? So to do that, we took advantage of a property of these dynamics where at some point these perturbations stop growing and you get into a steady state. And in this steady state, what you have is that the probability of getting a spike on the input, an extra spike or an extra missing, a missing spike on the input, has to be the same as the probability of getting an extra spike on the output. And we can estimate these in 2 different ways, and by equating them, we can have some estimation on the noise. In our case, it's kind of a random walk argument, but the bottom line is that we have something around 2 to 4.5 millivolts fluctuations between trials. Okay, So as I told you, the fact that you have fluctuation doesn't mean that you don't have, you can't actually have spike timing accuracy. All it means is that if you want to make the neuron spike at a certain time point with a certain precision, you need to make sure that you have a voltage excursion that is larger, say, than twice the standard deviation of the noise. So in this case, it would be something like 9 millivolts within, if you want it within 2 milliseconds, you need an excursion of 9 millivolts within 2 milliseconds. And then we did an experiment actually asking ourselves, how often does this happen? So we just take another neuron, or many neurons, we hyperpolarize them, and so they don't spike, and then we just count how many transients do you have of a certain size at a given time difference. Doing the statistics on that, we concluded that if a 1 millisecond precision event happened for layer 5 pyramidal neurons in our experiments, something like every 3 hours. So it's one spike within 25 milliseconds, 25,000 spikes, meaning that this seems not to be the major way that spikes are used in the cortex. If you go to higher, so lower precisions, then you start getting more reasonable estimates. Okay, so the conclusion of this part is that one extra spikes cause more than one extra spike in the network, and the perturbation grow and they grow fast, that the cortex seems to work in a noisy regime, that spike timing accuracy is not consistent with the rate of these excursions. On the other hand, we know that the cortex works, so it must use some other kind of model.
Can I ask for the previous slide?
This one here.
Yeah, so again, these are measurements. This is what? This is slice in vivo.
No, this is in vivo,
In vivo,
anesthetized rat.
anesthetized.
Sorry? It's anesthetized rat.
There is some input.
So in this case, we did, okay, so this is going to the next slide, not the previous slide. No, I'm going to talk about it in a second. But it's a weak point. It's a negative result, right? If you look for transient and you don't find it. So it doesn't mean anything, and of course
it's good result. But
what?
It's good result.
It's
You
in
say
one
that
given
the fluctuation,
condition.
the noise don't cause this excursion. Or
Right,
if
I
I want
mean-
to put something in it.
So first of all, it is an anesthetized rat, and
also
we did give some sensory stimulation. It's barrel cortex. We gave it a rotating drum, and it didn't make any difference. But I can tell you, although I cannot show it to you, I plan to show it. We're doing it now in awake mice. It doesn't seem to be very different, and the next step is obviously to do it during behavior. Maybe there you'll see something different. I'm going to talk about it in a second. So, okay. Okay, so I just want to contrast that. So this is the conclusion of that part of the talk, and I wouldn't like to say it. I don't need Chaim to criticize me. I'm criticizing myself here before, and I want to kind of challenge. Exactly. I want to challenge these results, and I'm the first one, I think, to say that there are serious issues with this kind of argument. So first of all, I want to present you a remarkable work that has been done by the group of Michael Fee, which shows us a completely different example to the one that we've seen in the monkey, where you have a zebra film, the recording from a nucleus called HVC, while the bird is singing a song motif which is repeated. And you see that on repeating trial, what you see here is recording from one neuron, the one neuron from HVC that projects to this other nucleus, RA, and this is like a SMA of a monkey, if you want. What you would see is that this neuron is emitting a series of bursts of spikes with amazing precision. So it's exactly the same time every time during the song. So we have an example, at least one example, where this neural system And this is not a sensory system. It's not depending on any feedback of the auditory system. It's a motor system generating a sequence with amazing precision. However, birds are not mammals, and they don't really have cortex. So one question is whether this system actually equivalent to cortex indeed. So one thing that I wanted to show you is work that we're doing now in the lab by two students, Nissim and Benjamin. We're starting to explore a mammalian system which is in the mouse. And what you see here is a male mouse in a cage. And here you see a spectrogram. So this is time and this is frequency. And we introduce a female mouse into the cage. And what you see in a second is that the mouse starts emitting vocalization, which are ultrasonic. You can't hear them, but I try to downsample them so you can see, you can hear what it sounds like. Again, complicated network but also this is what we're looking for. We want to see if there's any cortical area that is responsible for that, and if there is.
So cool. Really.
What?
So cool to an animal.
Why cool?
Taking him out.
Taking her.
Is this
You're talking about. You're talking about. Okay, this is what it looks like, the sonograms of that, and you can see that these are quite complicated temporal structure. So we hope that this will have another model that we can test temporal precision and the generation of temporal sequences in the cortex. Hopefully we'll find the cortical area that is responsible for them. But as Chaim said, there are two major issues with, there are many issues with what I presented you before, but two of them I want to address now. One of them is, of course, the anesthesia. Cortex is not doing anything during these experiments that I've shown you, so maybe it's all just because of the cortical state. The other thing is the assumption that we derive this noise estimates, by assuming linear integration, and we have many examples today to show that dendrites where synaptic integration takes place are very nonlinear. So maybe that also has something to do with that. So I want to continue with a project that is running currently in the lab. All the work that you're going to see is by Christina, a very talented scientist working as a postdoc in the lab. And soon enough, she'll probably be on the job market. So if you're interested, you can pick her up and ask her. So this has to do with IH, dendrites, gain modulation. And here how it that's the story. So what if you want to summarize everything that I've told you till now, the conclusion is that if you have high gain in the network, you're going to end up with a high level of noise. So the question is this something fixed, or is that something that we can somehow control or modulate in any way? And I want to present one direction. Of course, there are many, but one of them, which has to do with these neurons that we're talking about. And these neurons, these layer 5 pyramidal neurons, are special in the sense that they actually build up as two different neurons. There's like the somatic region, which is one neuron, which receive feed-forward input, say in somatosensory from the thalamus, for example, or from other areas. And there are recurrent inputs coming from its neighbors. Most of them are concentrated in this area. And then there is the apical tuft, where these neurons receive inputs from feedback higher order cortical areas, for example, or layer 2-3 neurons. Now it turns out that the gain, and now I'm using gain in a completely different way, but obviously related, the gain of the dendrites is very different than the gain of the somatic region. So for example, this is work done by Matthew Larcombe, injecting step currents into the into either the somatic electrode or the dendritic electrode with fluctuating current, similar to Mehring and Sosnovsky. And what you see here is that the somatic region behaves like a threshold linear neuron, where you have a threshold, and then from there on you have a linear increase in the firing rate as a function of the mean input current. In the dendrites, however, the threshold is shifted way to the right, so that it's much higher threshold. You need to inject much more current in order to reach spiking. But once you do get spiking, the gain is much different. It's much steeper.
So the spikes are somatic or?
The spikes are all somatics. Yeah. And if you look at the difference between them, what happens is that when you inject this noise in the fluctuation in the soma, you get individual spikes. But when you cross the threshold in the dendrites, what you get is bursts of spikes that are riding on top of calcium spikes in the dendrite. Okay. So what it means is that if you look at this neuron, you have like one gain to the somatic region and a different gain to the dendritic region, and also a different threshold. Meaning that if we were doing our experiment by injecting the current into the dendrite, maybe we wouldn't actually get these 28 spikes per spike, because this small current wouldn't make any difference to the probability of getting any spike to the network. On the other hand, it might be that the dynamics in the dendrites are such that these are also very close to threshold, and a single EPSP will get you over the calcium spikes, and you will get 3 spikes per spike. So we don't actually know that for now. But I want to continue with another feature that these dendrites have, which is that in this region, it's not only calcium, voltage sensitive calcium current, there's also another type of current, which is called IH. This is carried out by these HCN channels. These are the small channels from the title. These are highly concentrated in the apical tuft. You can see here by recording from Martin Kohl, that was doing cell attach recording on the dendrite, and there's like almost exponential increase in the density of these channels. And it turns out that these channels have interesting interplay with this calcium transient in the dendrite. So if you are in control, normal condition, and you stimulate the dendrite, you can get some calcium transient. However, if you block IH with this drug called ZD7288, what happens is that these calcium transients become longer and more prominent. So IH seems to be controlling the calcium transient in the dendrites. So the logic line here, which is not so logic, but anyway, the dendrites, the dendritic IH might control the calcium spike in the dendrite, which in turn are controlling the dendritic gain, and now the gain has something to do with the noise. Okay, so this is a loose relation between what we started and where we are now. So these are recordings that Christina has made from, and in this case, this is a awake mouse. Again, barrel cortex, layer 5 pyramidal neurons, and you can see when you inject step currents into the neuron, you see that you have IH in the dendrite. This is a signature of IH, sag current, and also this, you can see it also on the repolarization that you have an overshoot. This is a clear indication of having IH in the neuron. So this is control. Then we apply ZD, the same drug, topical on the craniotomy of the mouse. And you see that the drug actually managed to block the current. You can see it also on the population. Reduction in the SAG after the application of ZD. But again, I wanted to talk to you about dendritic gain, and we're recording in the soma, so for you, those of you who haven't picked it up, this is our somatic recording. So how can we say anything about the dendrites when we're recording the soma? And for that, we're using a very interesting trick from Matthew Larkham again, who showed, so this is an example from a slice preparation, and he's taking okay, so the trick is the following. You inject a train of spikes into the soma, okay? And these spikes are backpropagating into the apical dendrite. And this is what they look like in the apical dendrite. However, if you increase the frequency of this of the train of these spikes, what will happen is that they will start summating in the dendrites, and at some point they will cross the threshold to start activating calcium voltage-dependent calcium channels in the dendrite. And what will happen then is that repolarization you see now is riding this calcium transient, and it's elongated, and it's hard to see here that there is a difference between this actually propagates to the soma, and you can see it at the foot of the action potential. But this is another example from an experiment that I've done with Matthew. And when you align the last spike one on top of the other, you see that there is some difference between the transients. And if you plot that, the value, for example, of the voltage here as a function of the frequency, you see that there is some nonlinearity here. And you can define a critical frequency. This is the frequency at which this spike summates to a point where they start activating calcium transient in the dendrites. So to do that in vivo in an awake mouse is a whole completely different story, but still it's kind of possible to do that. So this is what we're dealing with, right? These are repeated trials, and we inject this train of spikes here, and you see that the whole mass, still if you align the spikes on the last spike, you can see that there is something here. And if you do it really carefully and only on the really good recordings, you can see that you have this effect also in vivo in an awake mouse, and you can plot this sigmoidal function of this area between these two recordings and as a function of frequency, and you can define the critical frequency. And we get this distribution of critical frequencies, which is in a way reasonable compared to what people have described previously in slices. But it's not far off. What?
Distribution among different cells?
Yes, different cells. This is about 14 cells, if I'm not mistaken. Okay, so the real question now is whether IH, as I told you and I tried to convince you, has any effect on this communication between the somatic region and the dendritic region. And indeed, if you block IH with this drug ZD, you have a decrease in the critical frequency from around 100 hertz to around 80 hertz. And this is, of course, doesn't have any significant, so in terms of we don't think that this protocol that we're using here has anything to do with the real role of how neuron behaves in vivo. It's just a trick for us to pick how much communication there is between these two regions of the neurons, and it seems that IH is able to modulate this communication, to modulate the gain of the dendrite, and how much the effect is so much. But mice are not running around in nature with that D in their pocket where they will put it on the head when they want to change the gain. So this doesn't mean anything physiologically. However, it turned out that IH was first documented actually in the heart, where it has a major role in the pacemaker of the rate of the heart. And as such, it's also been shown that it's sensitive to adrenaline. And it turned out that also in the cortex, there is high expression of adrenoreceptors, in this case alpha-2A adrenoreceptors, which are co-localized on the dendrites next to this HCN channel. And what people believe is that, although it hasn't been shown directly, that activation of these alpha-2A norepinephrine receptors inhibit cyclic AMP, which in turn modulate IH. And this is because these HCN channels have on the C terminals have binding site to cyclic AMP. And what seems to happen is that if you lower the levels of cyclic AMP, the activation of IH will shift to the left. Now, the sharp eyes of people of you will notice that the activation curve of IH is actually reversed to what we usually see, This is a very special current that is actually activated with a hyperpolarization. But in our case, what it means is that around resting membrane potential will have now much less IH active than we usually do. Okay, so does that actually work? So we're using a drug called guanfacine, and guanfacine is an interesting drug in the sense that it, first of all, it's an agonist of this adrenoreceptor, alpha-2A, specifically agonist of alpha-2A adrenoreceptors. But it's also interesting because it's a drug used for ADHD, ADD patients, people with attention deficit disorder. But in our case, what you can see is that if you apply the drug and compare control to guanfacine, you see that there is a reduction in the SAG between these conditions, and at the same voltage that you will get activation of IH. In control, you get very little activation of IH under guanfacine. Of course, if you shift it lower enough or hyperpolarize it strongly enough, you would start seeing IH as well. Okay, so again, does that has anything to do with the communication between the somatic region and the dendritic region? And indeed, again, this is very preliminary. It's not many neurons, but it does seem to be significant that you again have a reduction from around 100 hertz critical frequency to around 80. David?
I'm just curious. I thought, why did you go after noradrenaline? Why not dopamine? Because
So that's an interesting point. I'll touch upon it in a second. I'm going to touch it here. So yeah, again, this seems to be significant. We still need to work on that more to make sure that we don't have anything wrong here. But the picture, the schematic picture that we're trying to we're thinking along is that under control conditions or under certain conditions, I don't know what control is in this situation, when you have a lot of IA channels open in the dendrite, it kind of cuts off the feedback input from the apical dendrite to the soma. And when you modulate these currents and close them, then you facilitate the communication between these two regions. You facilitate this dendritic calcium spikes, and the feedback input becomes much more influential, also leading to a change in the output spiking of the neuron. And if this is true, we first of all needs to support it with some other kind of evidence. For some of them will be calcium imaging in the dendrite. We're currently working on that, but I don't have anything to show you which is reliable. So calcium imaging in the dendrites with this pharmacological interference, as well as a project that we have with Ian Duguid from Edinburgh University to try to activate endogenous fibers that carry norepinephrine in the cortex. But what is more interesting is try to see if we can, and this kind of connects to lines of thinking that are very prominent here in Jerusalem about the role of feedback from higher order areas and how they actually affect primary sensory areas, like the work of Merav Achisar, for example. Try to find a behavioral modulation, a behavioral task that will rely on feedback input or some, how would I say that? Some kind of higher order areas input to the neurons and change the way they behave. And this is work that we're doing in collaboration with Matthew Larcombe and with Jörg Winter from Berlin, from Humboldt University in Berlin. And obviously we want to do some modeling both on the single cell level and on the network of that work. So just to touch upon David's question, It actually been documented recently that there are also co-localization of dopamine receptors, which are doing exactly the opposite thing in that term. So what dopamine would do is actually increase the level of cyclic AMP and will actually increase the probability of IA channels to be open. And in some sense, I don't know if this is related in any way, but it could be that you have some competition between dopamine and noradrenaline on the levels of how much IH is expressed and is being opened in these dendrites. So this is obviously an interesting direction, and dopamine is related to ADD just as well, because the major drugs for ADD are dopamine reuptake inhibitors, like Ritalin, for example. So it might be related in some sense. Although all these works are concentrated on prefrontal cortex, and we're actually working in somatosensory, so I don't know how much it's related in this direction.
But
Anyway.
the fibers, I mean, the fibers from subsection nigra go throughout cortex. It's
Right.
It's not just focused in.
Yeah, sure. Okay, so just to conclude, I want to thank the people that are involved in this work. In London, it was Peter Latham. In the first part of the talk, Lisa Biren, who was a PhD student in the lab, Andros, and Mike Hauser, who was a PI in the lab. I did that work. And here in Jerusalem, it's Christina, responsible for the second part, and Nissim and Benjamin, who are working on these mice vocalizations. And I want to thank you here.
The IH has a big effect on the resting potential.
It does.
So I'm wondering if you counted in terms of firing rate, but if you counted in terms of the amount of current needing to activate that firing rate, is it the other way?
You're talking about the work of Matthew, for example,
You.
of our.
Yeah, I mean you used the firing rate of the cell as your metric, and then it went down. It's critical firing rate.
No, this is critical frequency. That this is not firing rate. This is like at what frequency do you get this shift in this in the
Spiking frequency.
at what? But it's a different thing. It's like at what frequency? Okay, let's just.
Frequency of stimulation. maybe.
Yes, exactly. It's the frequency of stimulation. Why is it? I turned it off. Sorry.
But you're inducing these back-propagating actions.
Right. So it's like at what frequency of this induction? So these spikes. At what frequency do you get this thing? Or better to look at it in this example. At what frequency of input do you get that? And for that, you just trigger the spike. So it doesn't. You might need you might need more current to do that, but it doesn't matter in that sense. But there is an interesting point there, which which bothers us for a while, and we still haven't completely resolved. So, in order to compare these results, we took a model that is by a student of Idan Segev, which actually captures this back phenomena, the fact that you have calcium spike in the dendrite and somatic. And then we just did the shift of the activation curve of IH. And what happened is that the hyperpolarization was so big that you actually didn't get any critical frequency at all, which is completely inconsistent from our experiment. So if you it doesn't matter how strong and what frequency you drive these spikes, they never get a calcium spike in the dendrites, which means that this model of the neuron was actually one of these models where you use genetic algorithm to fit different parameters. And since this
that's
was
what I'm talking about. IH has this huge effect on the resting potential.
but
and you
it
need
doesn't
some
seem to
magic
be
to compensate that.
but it doesn't seem, at least in our case, in our hands, when we apply this drug of contrasting, we almost don't see any shift in the resting memory
That's what I think.
potential.
There's something else going on.
Yeah, so we're not sure exactly what that is, but it's a valid question.
I'm just saying the other thing that's going on might be what's doing
Right,
it.
it could be related.
Yeah, so my question goes to the first part of the talk where you showed that you need to have a certain steepness in the voltage in order to evoke an action potential in the cell. So
Not to evoke it, but to evoke it with a certain precision.
Yeah, with a certain precision.
Right.
Yes. So I was now wondering, considering the fact that the conductance composition of neuron types is highly different, you would expect that if you would look on different types of neurons, that this relationship is very different, right? It depends on whom you are examining.
Yeah,
so
of
in
course.
other words, which type of neuron you are looking at.
Right. So it's true, and we looked only on 2 types of neurons, which was layer 5 pyramidal neurons and layer 2, 3 pyramidal neurons. Of course, it will be very different for stellate cells and others, but it could also be that the noise of these neurons would also be much larger, because if they have, for example, much higher input resistance, then every extra missing spikes will make a much bigger fluctuation. So it might be that it's scaling. It scales. I don't know actually. We never actually looked at that very carefully, so I don't know.
Yes, last question.
Again, to the first part, I mean, in a way, this experiment of perturbing spike of one neuron is not correct, because you are perturbing within the noise level of the network itself. So, but maybe a better experiment would be to think of a volley of spikes arriving at this neuron, and then looking at how this volley, something like Alex Reyes did, but maybe in vitro. And if you were to do that kind of thing here in vivo, then what happens?
Okay, so this is actually, let me, it's a last question, so I don't have much time to do that, but I want to show you that we actually were thinking along these lines, and we wanted to do it during behavior, and the method that we wanted to do that with was here. So this is a thing. Okay, so we were in London. We haven't started doing that here, but it's on the plan. The idea was to take using single cell electroporation you can target say 10, 20 neurons and introduce channelrhodopsin into these neurons specifically. You know exactly which neurons they are. I'll jump the movie, and you can actually show that later on you patch these neurons and you can actually activate them. And then we combine that with, we combine that again with multiple recording site electrodes, and the idea was that you activate a volley, you activate a bunch of neurons, and you want to see what happens in the network. So this is on the plan. One of the things that are difficult are actually to know that you introduce this probe very close to the neurons that you are in the same location without actually affecting them. And actually during the presentation that was here before, I don't remember the name, but there was someone here who was working on these polymers recording electrode. One of the things we're looking for is electrodes that will be transparent, and you can actually use them under 2-photon microscopy and kind of direct them into the location where you are doing the electroporation. So that's it.
Okay, so thank the speaker again. The next speaker will be Marlene Bartos from BLBT, that will talk about distance-dependent inhibition, a new mechanism of facilitating neural network.
Yes. Yeah, thank you very much for the invitation and to be here. The last time I was in Jerusalem, in Israel, is almost 25 years ago, and I'm visiting yesterday the city. I decided it would be a good idea to come more often to Israel, as well as specifically to Jerusalem. So I'm giving today a talk about unpublished data on distance-dependent synaptic inhibition, a new mechanism facilitating focal neural network oscillations. So rhythmic activity patterns or network oscillations are a hallmark of normal brain functions and can be recorded and found in various cortical but also subcortical areas. And although they are generated in a very broad frequency range, I would like to focus your attention today on the gamma oscillations, which are generated between approximately 30 to 120 hertz. I'm focusing on those rhythms because they seem to be the basis of cognitive functions such as sensory processing, but also enhanced attention, learning, and memory. And more interestingly, these rhythms seems to be disrupted in certain diseases, for example, schizophrenia, in which patients suffering from cognitive disorders. So here in the middle you see a human electroencephalogram recording, and shows you gamma activity patterns which have been recorded from the human brain, and they're characterized by this very rhythmic, regularly occurring voltage fluctuations, which indicate that the underlying rhythmic activity of neuron populations emerge in a highly synchronous manner. So we are mainly, in my group, we are interested how these rhythms are generated, but also how they contribute to behavior and cognitive functions. Now, in my previous work, but also in other in vitro as well as in vivo studies, and also theoretical studies, it was shown that these rhythms are generated by the synaptic interaction between GABAergic inhibitory neurons, shown here as blue circles, and glutamatergic excitatory principal cells, shown here as gray triangles. Most importantly, the properties of these inhibitory output synapses seem to be very important determinants of the frequency and synchrony of these activity patterns. So, for example, we have shown in the slice preparation of the hippocampus that the synaptic communication from interneurons onto principal cells has very specific properties, namely the inhibitory signal is quite strong and rapid, and based on theoretical work, which is summarized here in this raster plot, we could also show that these synaptic properties define or are important for synchronizing neural networks in the gamma frequency range. Now, these investigations have been done in the rodent hippocampus, which is highlighted here in this schematic, and I have drawn here a horizontal cut, and we are looking on the surface of this cut to show you the anatomical but also functional organization of the hippocampus. Now, the information and multimodal information is fed from the enteral cortex to the data gyrus or DG, and then transcoded into a sparse but also orthogonalized code for the subsequent CA3 area. And once the information is processed through the classical tree synaptic pathway, it is fed back to the cortex. So we are working on the hippocampus because this area is important for spatial navigation, and here in this area, including the denta gyrus, these activity patterns can actually be induced in a quite reliable manner, both in vitro as well as in vivo. Now, I introduced you to these GABAergic neurons. They form actually only 10 to 15% of the brain and nerve cell population, but several lines of evidences, for example, in vivo, but also optophysiological studies have shown that once you functionally knock them out, gamma rhythms are lost, or once you optophysiologically, for example, recruit them, the rhythmic activity pattern, so the power of gamma oscillations, goes up. And in this, GABAergic cells are highly diverse in their functions, and in this slide, I would just simply give you a glance on the morphological diversity. Do you have a question?
Yeah, just a short one. When you're talking now about the gamma oscillations, these are measured by in single neurons or in what?
As I said at the beginning, either EEG recordings or recorded under in vivo conditions. So in general, gamma activity patterns, this was like an introduction, yes? I'm coming now to the real data, but the introduction is based on in vivo recordings in rodents, or in this case in the hippocampus of these animals, or at the beginning I showed EEG recordings from the human skull.
So you refer it as the same phenomenon?
Yes. Mm-hmm. So here I would just simply want to show that interneurons are highly diverse, and this is their morphological diversity. So these are drawings from various types of GABAergic cells which have been labeled intracellularly. And we are mainly looking on one particular type, which I would highlight here in blue, which we call the basket cells, which have a very interesting morphology, namely they have very dense axonal avarizations which cover large areas of the principal cell layer, which are shown here in gray. And this organization is shown here schematically again. So we have here in gray the principal cell layer. Here I've drawn some somata of these principal neurons, which are called in the denta-gyrus primary cells, therefore here the abbreviation GC. And representatively, I have shown here, I'm showing here only one of these interneurons, one basket cell. So these neurons form inhibitory synapses close to the soma of the target neurons and are therefore able to control the timing, but also the frequency of action potential generation in the target neurons. What is also very important to know is that these neurons, and here you see again an intracellular labeling of such a basket cell, have this very dense axon here in the principal cell layer, and therefore make synaptic contacts to thousands of principal cells, but also interneurons. And thereby they can pace or control the activity actually of large neuronal populations. So this area in some preparations can even span the entire principal cell layer, which in the mouse is about like 1 millimeter length. So based on our studies, we have shown that the inhibitory output of such an interneuron is, as I said, quite strong and rapid. And this fits very nicely to in vivo observations now in the hippocampus, which showed that gamma activity emerges globally in the hippocampus at a frequency range of 30 to 70 Hertz, which was called also the lower gamma frequency band. However, in addition to this kind of global gamma oscillation, very recent in vivo data by Jackson and Williams have shown that in addition to that, like focal gamma centers at a higher frequency between 70 up to 120 Hertz emerge. And it was moreover indicated that these focal high frequent gamma activity patterns are generated by microcircuits, specifically by local synaptic interactions. So we were wondering how these interneurons can serve both. On one hand, the global synchronization of large neural networks, and on the other hand, can also serve the local synchronization of small microcircuits at the upper gamma frequency range. So one solution to this problem could be that this inhibitory output, as shown here schematically, is not uniformly distributed and the same on all target neurons, but it might be very highly non-uniformly distributed. So for example, if you look on the morphology of these neurons, you see the density of the axon is here in the center very high, whereas to the periphery it declines. And when you reconstruct the axon, you can indeed see that there is an exponential decline in the axonal density. So one possible solution to the problem could be that we have here a high synaptic strength at close neighbors which are innervated by these neurons, but that we have a very weak inhibition at more distant neighbors. So whether this is indeed the case and whether this might be a solution to this mentioned problem was the main question which we addressed experimentally. So how did we do it? So we were working in the slice preparation now, and what we did, we performed paired recording. So we evoked a single action potential here in one of those interneurons, and recorded the inhibitory current in the target cell. And this would be such an inhibitory current here shown in the gray trace. So we removed then the pipette and systematically searched for target neurons from the very same neuron and systematically increased the distance and tried to get as far as possible to the periphery. So here below you see another example from the same presynaptic interneuron, but now we recorded from a closer spaced principal cell, and you see that this inhibitory current has a completely different size. So weak inhibition here and strong inhibition there. So moreover, when we peak normalized these two currents and superimposed them, we also found that the time course between the two signals is different. So as a measure for distance, yes, between this neuron and that neuron, we use the synaptic latency, which is the time between the onset of the action potential and the onset of the inhibitory current. This was just simply based on the assumption the longer the action potential needs to get to its target's neuron, the longer should be the latency, right? And this we, of course, tested first, and this is shown here in this plot. We just simply measured the distant intersomatic distances in our paired recordings and plotted them as a function of the measured synaptic latency, and you see that there is a very nice linear and significant relationship, so we can use the synaptic latency as a measure for distance. So first of all, we measured the synaptic strength, which is plotted here as a function of distance or synaptic latency, and found that there is an exponential decline in synaptic strength. So in other words, we have strong inhibition at close neighbors and weaker inhibition at more distant neighbors. And this relationship could be best described by this coefficient alpha. Next, we took a look whether also the time course is different, and what we did is we just fitted this decay of the inhibitory signal with an exponential function and plotted here the time constant tau as a function of synaptic latency or distance, and saw that there is a linear relationship which could be described by the factor beta. So in other words, we have a fast inhibition at close neighbors and a slower inhibition at more distant neighbors. When we looked now on the very rapid component here, which I call the rise time, there actually we did not find this relationship as shown here. So in summary, inhibition, as shown here schematically, seems not to be uniformly distributed in this network, but shows a strong non-uniformity. We find strong and rapid inhibition at close neighbors, but weaker and slower inhibition at more distant neighbors. And this we found not just in principal cells at target neurons, but also for postsynaptic interneurons. And due to time limits, I can't show you all the data, but simply would like to summarize these data here, that distance-dependent inhibition seems to be a general rule which is independent on the nature of the target cells. And I think at least for synaptic strength, this also has been shown for the cortex. So now we were interested, and I would like to go a little bit more in the mechanistic detail. We wanted to know what is the underlying mechanism for this distance dependence in the strength and the distance dependence in the time course of inhibition. And one very simple explanation, so the first slide shows you what could be the mechanism for this distance-dependent decline in synaptic strength. So as I said at the beginning, when we have a very dense axon in the center and more weaker or less dense axon in the periphery, one very simple explanation could be that the number of contact points between an interneuron and its target cells just simply declines with distance. And if this is the case, then the probability that an action potential does not evoke an inhibitory signal should be at very distant neighbors very high. And this is exactly what we found. We measured the percentage of failures, and the failure rate increased with the synaptic latency or with the distance. So next we were wondering, so how many contact points are actually generated at close and at more distant neighbors? And this we did with kind of mathematical analysis, which is a binomial analysis, and for that we measured like 500 to 1,000 single inhibitory currents for a given paired recording, and did peak amplitude histograms. This is shown here in gray. This is an example for a strong synapse at a close neighbor, and this is an example for a weak synapse at more distant neighbors. And we fitted these data with a polynomial function in which various synaptic properties were kept as open parameters, most importantly n, or the number of release sites. Yes, and what we found was that indeed the number of release sites linearly increased with the size of with the inhibitory strength, and we found that approximately 10 to 15 release sites are formed for strong synapses at close neighbors, but 5 to 7 release sites are formed for more distant neighbors. So in summary, the reduced number of contact points here underlies the observed distance-dependent decline in inhibitory strength. So the next question would be, what could be the underlying mechanism of the slowing down of inhibition? And a very simple explanation could be that the composition of the postsynaptic GABA receptors differs. And here schematically you see such a GABA receptor, which is a pentamer, and the alpha subunits determine the kinetics of these receptors. And this is shown here. These are data from Rüscher, who found that in principal cells of the hippocampus, alpha 2 more or less is the underlying, defines more faster, and alpha 3 a little bit slower time courses of GABA receptor mediated currents. Yes. So what we did, we labeled a single basket cell, this is shown here, defined an area close to the soma and a more distal area, and looked on co-localization. So we looked on the synapses, putative synapses, and used antibodies against this alpha 2 and the alpha 3 labeling, and looked on co-localizations. And to make a long story short, we found that the co-localization for alpha 2 declined with distance, but for alpha 3 increased with distance. These are only few data points, so we don't at the moment have significant differences. I'm talking here about trends. This needs further verification. But these data indicate that the different composition of the GABA receptors may underlie the observed distance-dependent slowdown of inhibition. So in summary, on the basis of the experimental findings, I have shown you that the functional properties of these interneuron output synapses are highly distance-dependent. They elicit strong and rapid inhibitory signals at close target neurons, but weaker and slower inhibition at more distant remote neurons. Secondly, distance-dependent inhibition is independent on the nature of the target cells, and therefore indicates that this is more a general principle of the hippocampus. And finally, I have shown you that the decline in inhibitory strength with distance seems to be caused by a reduced number of contact points, whereas a slowdown of inhibition appears to be due to a change in the postsynaptic GABA receptors. But now I would like to come to the two important questions regarding the functional contribution of distance-dependent inhibition. Namely, first of all, we ask, does this form of inhibition support the mentioned global gamma oscillations in the hippocampus at the lower gamma frequency range? And secondly, does it contribute to the synchronization of small cell assemblies at the upper gamma frequency range? Now, in order to address these two questions, we performed network simulations, and in this slide I would like to introduce you to the single cell but also to the network model. So the neurons were modeled as single compartments with Hodgkin-Huxley conductances to reproduce these fast activity patterns in the interneurons and more slower and accommodating discharge pattern of principal cells. And we modeled the synapses, actually based on our own experimental findings. The connectivities were also based on anatomical studies from our own group, but also from the Burgard lab. So the network consisted here of 200 interneurons, shown here in gray, and 800 principal cells, shown here in black, and thereby reproduced a numeric relationship between the two types of neurons as found in the real brain. They were randomly connected, and what we did is these neurons were located on a circumference, and the distance between the neurons was about 50 micrometer. This was important because every signal which was sent from one neuron to the other arrived at the target neuron after a given synaptic latency. And this latency was defined by the distance and, of course, the speed of action potential conduction. So we had two models, and in the following, the color code stays always the same. We have in blue the non-distance dependent network, yes, in which the synaptic strength was uniformly distributed in the network, and also the time course of inhibition was uniformly distributed. And the red one is the distance-dependent network, in which the synaptic strength declined exponentially with the coefficient alpha, defined by our experiments, and the time course of inhibition increased linearly, defined by the coefficient beta, also defined by the experiments. And most importantly, the synaptic latencies were kept in a realistic range. Yes, not that we have, like such as Aertsen said, yeah, too long latencies. So the first question is, does distance-dependent inhibition support the kind of global low-frequency gamma oscillations? And this is shown here in form of raster plots, where we plotted these 800 principal cells as well as the 200 interneurons as a function of time. And I mean, you are very likely very much familiar with raster plots, but I still say it. Each of these dots indicates when such a neuron discharge an action potential. And in blue and red we see the interneurons, and in black we see the principal cells. So what we see here, that if such a neuron network was activated by Poisson trains of excitatory inputs, both networks generated globally gamma oscillations at the lower gamma frequency range between 50 to 75 Hertz. This of course depends very much on how you drive the network, yes. This I'm not going to show today. And most importantly, the principal cell activity was quite sparse, so similar to in vivo investigations, indicating that indeed such a network with distance-dependent inhibition supports the generation of this global gamma activity. So next we ask the question, so can we introduce, so how is it, can we now introduce on top of this global gamma rhythm a focal recruitment of neurons at the upper gamma frequency range? And this we did in the following manner. We applied a focal additional excitatory drive onto a subpopulation of neurons, which resulted in a marked recruitment of both interneurons and principal cells, as shown here in the middle. However, several differences emerged, and those differences I would like to highlight now. And what you will see now is that these squares here are shown at higher magnification here now in the middle. So we see in black the principal cell activity, and again in blue and red the interneuron activity, non-distance dependent network, distance dependent network. So first of all, we saw that the average discharge rate in both the interneurons and principal cells was here in the DD network much higher than in the non-distance dependent network. Secondly, we found that the activity was much more phasically modulated here in this red versus the blue network, which resulted in a significantly higher power of the activity here for both the interneurons and principal cells in the distance-dependent network, but much weaker power here in the non-distance-dependent network. Moreover, we found that the activity was really tuned to the upper frequency range, in this area, case something like 85 Hertz, whereas for the non-distance-dependent network there was a much broader scatter in the frequency. So this indicated to us that the underlying inhibitory signal seems to be much more phasically modulated than in the non-distance dependent network. And whether this is indeed the case, in order to address this, we computed the local feed potential analog from this network, and this is shown here as a black trace, and indeed it shows that the local feed potential activity in the circuitry is characterized by a much stronger phasic modulation than in the non-distance dependent network. So in summary, with this slide I would like to show you that distance dependent inhibition seems to support the generation of global gamma rhythms and the synchronization of focal cell assemblies at the upper gamma frequency range. Now the next question which we addressed is, so how does it do, so what is the difference? So how does distance-dependent inhibition support this facilitation of these focal gamma rhythms? And in order to address the question, we separated the two types of synapses interneuron synapses and interneuron principal synapses, and took a separate look on them. And what you see here on the left-hand side is the simulation work for only the distance, for the distance-dependent inhibition among interneurons. So what we did, we picked out one neuron from this network and simply took a look what is the total inhibitory signal or total inhibitory conductance that a single neuron sees by all of its convergent inputs. And this is shown here. And what you see is that in the center, interneurons receive a very phasically, strongly modulated inhibitory input, whereas in the non-distance dependent network, this amplitude, it's less phasically modulated, resulting in a much higher power of the signal. Interestingly, in the periphery, the situation is completely different. In the periphery of the network, due to the slowing down of inhibition and weakening of the inhibition in the periphery, the inhibitory conductance was much smaller and less regular, resulting in a marked reduction in the power. So it indicates that there is a high contrast between the center and the surrounding network. So how about the what is the functional role of distance-dependent inhibition onto principal cells? So in order to address this question, what we did now, we just simply paced the interneurons, so they were always active at the very same time as shown here, and took here only a look on distance-dependent inhibition onto principal cells. So what we found is here, when you look just simply on the scatter of the principal cell activity, we found that in the distance-dependent network, the activity of the principal cells was much more phasically modulated than in the non-distance dependent network. So this indicated to us that very likely the underlying inhibitory signal is much more phasically modulated, and indeed this is the case. So when we looked on the total inhibitory signal, which is shown here, and superimposed it with the inhibitory signal in the non-distance dependent network and overlaid them, you see that in the distance-dependent network, the signal is much larger and has a much more, phasic appearance, or much sharper appearance than in the non-distance-dependent network. And so we believe that this sharpening of the inhibitory signal is the main factor which drives the circuitry. So in summary, the high strength and the phasic modulation of distance-dependent inhibition improves the local synchronization of interneurons, and at the same time it improves the entrainment of principal cells, and thereby results in this very high focal, high power of the focal gamma oscillations. Yes?
How do you explain the fact that we have like slower and weaker synapses on the periphery, so we have much more phasic and larger?
So under these conditions, when you recruit this group of neurons, yes, they of course have axonal arborizations also to the periphery, and thereby conduct the input also on the periphery. And since the inhibition declines with distance, is getting weaker and weaker, also the inhibitory signal, so the pacing signal, is getting weaker but also broader. So our assumption is that we have then in the center, due to very short, you have like a small group of neurons which is activated in the center. You have short latencies and very strong inhibition, which supports the very focal synchronization of neurons. Whereas to the periphery, I would say you have like a more slower and more kind of tonic inhibition to the periphery, which loses its, let's say, pacemaker-like phenotype, and which results in the fact that these neurons are not precisely timed in the activity, but that there is a broader jitter, and therefore you have like a lesser synchronization in the periphery.
So that's what I saw.
Yes.
This figure on the right shows
Yes.
exactly the opposite. No? I mean, is that in the periphery or is that in the center?
And so, the problem is if you want to, we try to dissect it here. So we are looking here only on interneuron, and here from interneuron on principal cells. And in order to really look only on this synapse from interneurons onto principal cells, we simply paste the interneurons, yes, so that they are always active in the same time in both circuits, in the non-distance and in the distant-dependent network, and then just simply look what happens. Here you have the situation where you have a homogeneous inhibitory output, and here you have the distance-dependent output. So you cannot really compare here center and surround. We just simply wanted to know what is the effect, yes? And the effect seems to be that here distance-dependent inhibition is a much better entrainer of principal cell activity, yes? But this comes now, what the interesting point is now, so since the activity in the center is highly synchronous and more synchronous than in the periphery of the network, we were now wondering whether this is a big advantage of the circuitry so that it can generate several centers and thereby enhances the computational power of the network, yes? So if a network is able to have several like focal gamma rhythms in which separately information can be computed, this could be an advantage of distance-dependent inhibition to improve its computational power. And this is exactly what we tested in the last slide, and this is shown here. We just simply introduced a second stimulus and took a look how the two local feed potential oscillations interfere with each other. And this is an example again for the non-distant and here for the distance-dependent network. And we computed the cross-correlation as a measure of interference. Yes. And the data are summarized here. So what we did, we systematically changed the distance between the two gamma centers. This is plotted here on the x-axis. And here we increased the amplitude of one of the stimuli. And then we measured how strong is the interaction or interference between the two centers. And this is shown here with the color code. So warm colors indicate a very strong interference, whereas blue colors indicate a very low interference. And the really big difference is that we found is that in the non-distance dependent network, independent how far away these two gamma focal gamma oscillations are, they interfered with each other. So one influenced the other, whereas in the distance-dependent network, there was no interference whatsoever, only if they are very, very closely spaced, as shown here on the left-hand side. So indicating that indeed the computational power of such a network could be boosted by distance-dependent inhibition. And this prediction we tested experimentally, and this is shown here on the very right side. So what we did is we put two electrodes in the slice preparation and puff applied high potassium solution and recorded the local gamma oscillations in the slice in the granular cell layer here. And the recording of the LFP signal from one electrode is shown here, on the second electrode here in gray, and measured again the interference between the two focal gamma centers. And what we did is we systematically again changed the distance between the two focal gamma oscillations and measured the interference between both of them. So the model predicted that in a non-distance dependent network, it didn't really matter how far you put them from each other, there was always an interference, whereas in the distance dependent network, the interference declined exponentially. And this are our experimental data. They indeed follow our model's prediction, namely the interference measured here as cross-correlation declined exponentially with distance between the two gamma centers. So in summary, these data, I think, show very nicely that several local gamma oscillations can coexist in neural networks with distance-dependent inhibition. So in summary, based on the computational work, I have shown you that distance-dependent inhibition enhances the synchrony of gamma activity of focally excited cell assemblies. It thereby enhances the contrast between the center and the surrounding network and reproduces the granular appearance of focal gamma oscillations which have been observed in cortical circuits in vivo as well as in vitro, including the hippocampus. And secondly, distance-dependent inhibition supports the emergence of several independent gamma centers and may therefore enhance the computational power of the network. So I would like then, at the end, like to thank my group, and specifically Michael Strüber, who did the experiments which I showed today, as well as the theoretical work during the course of his now finished PhD thesis. And at the end, I would also like to thank some collaborators, but specifically Peter Jonas and Josef Bischofberger, with whom we had several discussions on the course during the course of this project, and which also helped us to focus a bit on our investigations. So I thank you very much for your attention. And yes, please.
So what might be the computational function of those gamma or independent gamma oscillations?
Independent gamma oscillations, yes.
You're saying that it increases the computational power,
Yeah.
but what kind of computation is carried by the gamma?
So the global gamma oscillations actually are generally thought as functioning as a more global context signal for information representation, whereas the focal gamma activity we consider as a focal drive of certain information which conducts one of the multimodal inputs which come from the enteral cortex and hit certain subpopulations of neurons in the cortical structure which encode this form of information, so that you have a kind of, you can kind of have different internal cortex inputs to the hippocampal circuitry, which are then independently processed or computed.
So the excitatory connections are not distant dependent?
This is a little bit of a problem. So when we talk, so wait a minute. So we are talking here about two different forms of the excitatory inputs. One from the principal cells, which are embedded in the circuitry. There we seem to have also a form of convergence, so very close neighbors of principal cells contact very local interneurons, yes. So there is a structure in the circuitry, yes. Whether it's distance dependent, I don't know. And yes, but it is clustered. There is kind of a clustering, yes. And then we have, of course, the external input coming from the enteral cortex. And how this is structured in the hippocampus, this is actually not really known, yes. So.
That strength of inhibition is very important, also in the non-dependent network. If you reduce just the strength of independent, can you get local computation in the non-dependent network?
If you reduce the inhibition.
The strength of the inhibition.
Yeah.
So it becomes more like more local network.
In the non-distant discipline.
Or what does it do also?
I mean, if you reduce the synaptic strength, I mean, there is a minimum strength needed in order to pace the system anyhow. So I would not say that you support this kind of focal gamma activity, this not, but you would lose at some point, at a certain value, you would lose the synchrony.
Is the strength of inhibition takes from the data?
Yes. Yes.
No more questions? Thank the speaker again. Last PhD session Kleinfeld, San Diego. We talk about breathing, the master clock for orofacial breathing.
Okay. Okay, let me thank Yifat, and it's a pleasure to be here. So I want to talk about some recent work in the lab that's come up, and there are studies of active sensing in the rodent fibrosis system. So let me just give you some of the essential background. We often think of sensation as a passive task. You sit, you show, if you do vision physiology, you show this. If you study the fibrosis system, your rat sits there and you shake the whiskers. But of course, this is not the way the world works. Our eyes are darting around constantly, yet in our mind's eye, we put together this somewhat stable picture of the world view, which means we have to know where the eyes are. As we touch objects around us, touch them with our feet, touch them with our hands, touch them with our fingertips, we're constantly moving, where the pressure sensors perhaps are getting the same amount of impact as we make contact with objects, and yet we put together this sort of stable picture in our mind's eye. So we have to know where objects are in space, okay, where our senses are in space, I should say, in order to interpret sensory input. So the point is that all sensory activity is really a combination of a motor act to tell you where your sensors are, or some abstract sense, some deformation of the body sheet, together with the input that's coming from the sensors. So this is sort of what's been driving sort of our thinking and research. And I want to describe today a problem in the vibrissae system as to how you actually generate the motor controls and how these motor signals, in fact, may act as a sort of carrier signal for interpreting sensory information. So I'm going to be doing all our experiments on the rodent vibrissae system. We suggest it has a number of great advantages. The rats sweep their vibrissae through space. As I'll show you, they do it with a sort of a scalar degree of freedom in the behavioral act. There's tremendous symmetry to the way they move. And further, they could be trained to do a task, and they could be trained to do these tasks while their head fixed. Okay, so there's a lot of advantages in the laboratory. So let me explain the work. This is primarily the thesis work of Jeff Moore I'm going to talk about, and it's work when Martin Duchenne was a visitor in our laboratory, and sort of ongoing work which relates to synchronization of rhythms by breathing is continuing work with Fan Wang at Duke University. Okay, so the title of the talk is breathing, and I'll show you how breathing gets into this picture, but let's start with whisking. Let me just get everybody up to speed and take you through a little travelogue on a review of whisking. Okay, so I know this is going to happen. This is the
snow.
It's the snow. I think this is the Bill Gates. Okay, Bill Gates tax. Okay, so this is the thesis work of Dan Hill, and there's about 5 different sets of muscles that are driving the vibrissae. It seems relatively complicated, and what Dan did was record from all these groups. I'm showing you here. This is the EMG, the electromyogram. It's the rectified muscle activity that for 3 of these 5 cells. Now the point is that you can imagine 5 neurons. This whole thing can be very complicated, but in fact you see their signals are just phase offsets of each other. And in fact what Dan did is he measured from the activity of all the muscle groups, and he looked at all the sort of elastic properties of the different muscle groups and the connection pattern. And then this is what's shown on top is sort of a very rarefied picture of this model, which is springs, which are the wiggly lines, the dampers, which I'm not showing, which are in parallel with all the wiggly lines, which is just damping, and force generators, which are the muscles, which are the eyes and the ends. And the point is that the very simple output motion can be predicted from just this simple rhythmic input, and that's what's shown here on the bottom. So what we do is we just take the EMG input and put it into this mechanical model, which is shown in the thick line, and the thin line is the actual measurements of the output, and the correspondences are right quite well. And the only 3 parameters some overall gain between neural activity and the constriction of the muscles. So the point is that the motion can be understood in a kind of seemingly simple way, that somewhere there has to be a clock, right, that's generating these signals. So it suggests to us, if we really want to begin to understand the motor act, which is driving the vibrissae that the animal is using to detect objects in space, we want to understand where this clock is.
These vibrissae, are they not independent? Do they always move like this, like you just showed?
They always sweep together, okay? Now, in the problem with biology, there's always the 1%. And so in one paper, once you can show that they could actually break this symmetry. But we'll be happy if we understand the 95%. And the 95% is that they move together, okay? Now, I started by saying that this is we claim that this is a very good system to study active sensing. So what that means is that the rat really has to know where its whiskers are in space if we're going to say we're interpreting sensory input, in the framework of motor control. So what this means in practice is if I teach the animal to talk to me and tell me where an object is in space, say a pin that's being dropped down in this position versus this more caudal position, the thing is, and the animal hits it with roughly the same forces, does the animal know that it contacted an object here versus here? Okay, so how can we describe this? This is the sort of thesis work of Summer Mehta. So that what we do is we train the animal that one of two pin positions are going to be associated with a reward, and we do operant conditioning to get the animal to come in and poke her nose into a nose poke, stay stable, and then we're going to drop one pin or another pin. One of these is rewarded, one of these is not rewarded. She has to talk to us. The way she talks to us is pushing a lever, and the frequency of lever of pressing is a way of telling us what's a reward if is expected or not, and if it's rewarded, she gets a little sip. So this is just standard operant conditioning. If you visit San Diego and you go to SeaWorld, Shamu will jump, and she's trained to jump. Every time she moves in the correct position, she gets a fish. She doesn't do it in the correct position, no fish. It's the same strategy with the rat. Every so often, operant condition fails, and the trainer gets stuck under water for half a minute. Not every rat is perfect, so be it. But the basic idea is this. We could look at the lever presses. So this is a raster plot like we've seen earlier, but instead of spikes, these are lever presses. And the idea is if you squint, I'm trying to convince you that the green dots, okay, which is a rewarded position, come earlier than the red dots, which is the unrewarded. So in fact, if we integrate, okay, over trials, which is integrating in this direction, and then I plot the integrals, we can see that the rewarded, the increase in lever pressing comes up quicker for the rewarded position than the unrewarded position. And these curves sort of break off from each other at around a half a second. We can make some arguments about the mechanical time, the motor time of the animal, and if you believe the arguments, we get down to about 100 decisions within about 150 milliseconds or so, or about the time about 1 to 2 whisks of the animal. So it's a pretty quick decision. Okay, but the claim is that this kind of experiment suggests that the rodent system, the whisker system, is a good place to study active sensing because the animal knows where the whiskers are. So let's now go back and let's look at whisking a little bit. And so what the animal does is she could whisk almost over a range of something like 160 degrees. But in reality, she picks a narrow range in space, sort of a region of interest. And the region of interest has some amplitude, and it has a sort of center point or midpoint. And this is a typical whisking bout. This is data from Dan Hill's thesis work. This is about 2 seconds of whisking. And the thing you notice, it's not a perfect oscillator, but it's close to being a very good oscillator. And this suggests perhaps that we could describe this, at least in a mathematical sense, as a rapidly changing phase, and a very slow change in amplitude. And that's just this is just a mathematical exercise to break this down into a rapidly varying phase as you go from cycle to cycle, and a slow change in amplitude. And of course, by construction, you can reconstruct it. That's the red curve versus the raw data, which is the black curve. And it suggests, though, perhaps that what nature is doing, okay, is actually having independent control over fast changes in position, like the fast oscillation, and perhaps some overall slow control in oscillation. So this sort of analysis just suggests that the position of where the whisker, or whiskers, the pad, are in space is some slowly varying amplitude times some rapidly varying phase signal, okay? And the question is, does this exist? And if it does exist, where might this fast signal be generated? Because that's sort of a carry wave for the sensory input that's coming in from the vibrissa. Okay, so what's the evidence that this kind of split, that nature chose this kind of split, because it didn't have to be so? So one piece of evidence comes from work done in Vogel's lab at the Weizmann Institute. What they did is they looked at whisking. This is a control trace where they put a systemically added an agonist for endocannabinoid. What happens is they realized that the agonist affected the amplitude of the whisking, but it really didn't change the frequency. And if they put an antagonist in for this receptor, all of a sudden the amplitude was very, very stable. But again, the frequency was not affected. So what the receptor is not the important issue, okay? The important issue is that they got a split between amplitude control and temporal control, because agonist versus antagonist changes the amplitude. This is just a cumulative probability distribution over many experiments, but it doesn't affect the periodicity of whisking. So this was one piece of experimental data that suggests that nature followed this course. Another possibility for whisking is that, well, there's motor control driving the position of the vibrissa. There are pressure receptors in the vibrissa. There could be a closed feedback loop. So maybe this closed feedback loop is actually forming, sort of a positive feedback network, sort of like taking amplifiers in a circle, they'll chase their tail, or like the oscillator in the leech for swimming, it chases its tail. In order to test that, we simply want to cut sensory feedback. So this is experiments done by Rune Berg, he's now a professor in Copenhagen. What Rune did is every time the animal whisks, he found out what the frequency was of whisking, then the animal would stop and whisk again, and you got some distribution of whisking. He transect, cut the feedback. Okay, the animal continues to whisk, very slight shift in the frequency of whisking. But another possibility is that actually you're using feedback in some sense to improve the performance of whisking as a serious feedback signal. Now these data were taken when animals were whisking for at least 4 seconds, if not more. So we could ask what the fidelity is. So if an oscillatory signal, okay, then the fidelity of the frequency is 1, over the total time of the whisk. Okay, so in the optics world, that's called transform limited. So it's a 4-second whisk. That means the frequency should be defined to within 1/4 or 1/4 of a hertz. That's called the Rayleigh frequency. And you can see that the experimental distribution of frequencies basically drops. The widths drop off at 1/4 of a hertz. You transect it, it also drops off at 1/4 of a hertz. So we don't need feedback. So this suggests there's a circuit that's sitting somewhere in the brain that's acting as a good clock. Okay, so where could this be? Well, 1964, Beatles performed in Shea Stadium, and Wally Walker did the following experiment in Wisconsin. Okay, this is sort of like the 10 plagues, right? This is an animal whisking with nothing happening, and now it's lost its nose, and now it's blind, and now it's deaf, and now all of cortex is missing, and now all of frontal cortex is missing. And speaking of ADD kids, and this is with Ritalin, okay, but the point is that no matter what the perturbation is, sort of experiments you would never get through an IACUC committee today, okay? The rat kept whisking, okay? So it's not in the cortex, it's maybe in the midbrain, it's maybe in the brainstem, okay? In rat. Okay, so let's hunt, okay? Let's take a hunt to find out where this oscillator is. All right, one clue comes from, this is recent work done by Sebastian Hedaro in Ehud Assaz's lab, is that there are muscles. The same muscle is often involved in breathing and whisking. And one particularly dramatic example of this is a traversus muscle, traversus nasi, which sits at the tip of the nose. And every time you breathe, it expands out rats are obligate nasal breathers. It expands out the size of the nasal cavity. At the same time, it pulls the whiskers back, okay? So already there's some clue that maybe there's some control structure that's common to both breathing and whisking. Well, Welker did another experiment back in '64. You take a rodent, a mouse, or a rat, and you perk it up. You maybe even in the laboratory, you eat an apple and just breathe on the animal, and she just starts whisking and starts sniffing very, very rapidly. She's all perked up. This was, again, done back in '64 by Welker, and what Welker found, in fact, is that sniffing, which is a rapid form of breathing and whisking, will completely synchronized. Okay, so we repeated what Welker did. And what we have is, this is a rat. It's head fixed. We're measuring vibrosposition with a camera. We're measuring breathing with a thermocouple. And breathing is shown in red. Whisking is shown in blue. So you can have breathing without whisking. And then you could have also whisking and breathing. And they're perfectly synchronized. So at this point, you might say, well, maybe it's just the breathing clock. Okay? A so-called pre-Botzinger center found by Jack Feldman. And there's just sort of a gate. Okay, but you can do something else. You can actually stop the animal from breathing. You can just give it a puff of pneumonia as a control, which I'm not showing you. You could be recording from cells in the breathing center and they shut down. Okay, so now this is whisking in blue without breathing. So this suggests, in fact, there are independent clocks, one for breathing and one for whisking. And the biggest clue came from having an animal which is not doing this sort of rapid breathing or sniffing, but actually breathing, just basal rate, slowly. This is 1 second. It's breathing at about twice per second. At the same time, she's whisking rather rapidly. And you can see it's whisk, 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 and it's kind of winding down. And then she breathes, and it's like a reset of whisking. So it's like whisk, 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 breathe. And you get this reset of whisking. So this data suggests that breathing, perhaps, is resetting the whisker motion. Okay, so this is 3 data sets. So we took something like 20,000 data sets, and what we're going to do is align them at the onset of inspiration. Okay, and that's when we measured with the thermocouple. It's at the red. It's this red arrow. There's a little bit of a correction from the time the neural signal comes in until you actually start to squeeze down on the rib cage. So that's actually, the breathing's actually starting here on this green line, and then each blue dot is when the animal whisks. So when you're whisking rapidly, or breathing rapidly, I should say, breathing and whisking are perfectly synchronized, right? The red bands for breathing and the blue bands for whisking are just 1-to-1. When you start to breathe very slowly, so you breathe and then you whisk and you whisk and you whisk and you whisk, and then finally you breathe again and it's reset, right? And then you whisk again. And the whisking here looks like it's completely incommensurate. It's running at its own frequency, and then finally it's reset. So this suggests to us the following picture, that there's a yet to be determined CPG, and that it's reset by breathing.
How do you know there's no common CPG to go? Because of the reset?
I mean, okay, so it sounds like turtles all the way down, and there's a common breathing center to the breathing center to the breathing center to the breathing center. So you could destroy the pre-Botzinger, which I hate to tell you happens by accident in the lab, and the animal will never breathe. Okay, anatomically, there's going to be no other inputs coming into this pre-Botzinger. And then I'll show you, we'll do anatomy experiments soon to show you that there appears to be no other input that, sort of necessity and sufficiency about the whisking center.
The breathing CPG is well identified, anatomically.
The breathing CPG was determined by, right, it's well identified. Okay. I mean, now.
The ammonia experiment in that sense is not conclusive, right? Just because they can voluntarily stop the breathing doesn't mean that the pacemaker is switched on.
No, okay, so I didn't show you here. I mean, we recorded from these cells. I mean, at the same time, yeah, I'm doing behavior first, but we have an electrode in the pre-Botzinger in these experiments. Okay, so it is turned off.
Okay.
I mean, needless to say, we did all these things with halo-rhodopsin, which takes 3 weeks, or you could do it 20 minutes with ammonia. So I'm sort of a minimalist. But, I mean, this is the knee-jerk reaction of every editor today. What happens with, XXX opsin? Okay. So the lesson here is that the pre-Botzinger respiratory complex, which I'll show you in a moment, is involved in this protraction phase of whisking. Okay, so now let's start to get into the circuit basis for the whisking pattern generator. So basically, we wanted to milk behavior as far as we could. So just to give you, by way of background, this is the follicles. The whisker is hanging off this end. There's a sort of a ring which contains pressure receptors. Okay, it's innervated by something called the trigeminal ganglion. This goes over into secondary sensory neurons in this long structure called the trigeminal nucleus. These details aren't very important. It's a rapid feedback loop over to the motor nucleus. And this motor nucleus, each of the motor neurons, okay, that drives the muscles, that moves the follicle, okay, gets input from at least 20 centers, right? So one question could be, is there a second pattern generator that somehow we didn't discover? Okay, so that I can't, that's open, okay? But we're hunting, right, for neurons that are premotor to this motor nucleus that generate this rhythmic oscillator. So we're down in the brainstem, and so let's proceed. So the first thing we do is we try to reproduce what was known from the past, from Feldman's lab. So we go to roughly the coordinates of the pre-Botzinger complex, okay, which is going to be sitting towards the ventral edge of the brainstem, this thing called the intermediate reticular zone, the medulla. And we have a unit here, and we can see it has a certain firing pattern. We're recording breathing in red, whisking with a camera in blue. You can see that this is breathing, and there are epochs where it's not whisking, but it's breathing, and the activity is very nicely synchronized. There are epochs where it's breathing very quickly, whisking very quickly and breathing slowly. So this just reproduces sort of old data that tells us we know how to find the pre-Botzinger complex. And I'll just mention this mystique that somehow it's hard to record from the brainstem, but it's a false mystique. You wind up the animal very carefully, it's head fixed, and then you could always get back to the pre-Botzinger complex. And that's oscillating, okay? And that's the zero for your coordinate system. And then you move from there. So we then just sort of march, okay, away from the pre-Botzinger complex, and we had some clues as to what direction to march. And we find another area, okay, in which, again, there's an epoch here in which breathing is very slow, whisking is much faster than breathing, and these units, okay, are well synchronized with whisking, and they're not synchronized with, and they're not locked to breathing per se. So for every unit we record, you make a mark with a die off to the anatomist, you slice up the animal, you put a dot, and you sort of map it on top of the anatomy, and you can see then where the cluster of breathing units are, and where the cluster in red, and where the cluster of whisking units are here in blue, right? So we sort of form a map of where the nuclei are. Okay, so we have this newly identified zone of neurons in the intermediate reticular formation. It's the word for the day. And now let's ask questions about sufficiency. Okay, so everybody knows there's a lot of miracles in neuroscience. I mean, the first one is that cells stick to glass. Another small miracle happened here. Cyanuric acid, which at high concentrations is the way to cause lesions, in low concentrations binds to the NMDA receptor and causes neurons to become active. So it turned out that it activates the CPG for whisking more rapidly than it activates other CPGs. So we could add cyanuric acid in the same location where, based on recording, we claim the Vibrissa CPG is. So this is measuring from the muscles. This is a lightly anesthetized animal. You see very nicely that the EMG, the Vibrissa position, are nicely turned on. So we added cyanuric acid. We get about an hour's, almost 2 hours worth of recording. The frequency just sort of slowly ramping its way down. Now the beauty is, because this is a lightly anesthetized animal, you don't have motion artifacts. Now we can go intracellular. So these are recordings at that location. And these are recordings of neurons that are firing in phase with a protraction of whisking. And these are neurons that are firing in phase, or rough, not quite pi out of phase with protraction or with retraction of units. Okay, so you get this kind of neurons that are sort of seesaw, some in phase, some 180 degree out of phase. But this is really a powerful tool then. It's not only proves sufficiency of this region, but it's a powerful tool to which you record intracellular by activating the CPG and the animals anesthetized. The other thing that's important is when we activate this, at this point in time, whisking and breathing completely break off and become incommensurate. Again, consistent with separate oscillatory areas. Okay, so this shows that this zone can produce rhythmic drive to the vibrissa, and let's see if this zone is necessary. So to see if it's necessary, you have to do a lesion experiment. Okay, and again, in a perfect world, you somehow identify these cells by some factor, some lineage factor. This we don't have yet, but we know where they are, so we can go in and we can lesion the region, and there's different ways to make lesions. We can make an electrolytic lesion, which opens you up to destroying axons of passage. We can make a viral lesion using Symbis virus that kills all the neurons, which doesn't have problems with axons of passage, but it takes a few days. So I'm showing you here is the data from one lesion. We've killed this center on the ipsilateral side. So remember, this is the brainstem. We haven't crossed yet. The contralateral side, the animal is whisking normally. Again, there's different ways to make lesions, and the bottom line is that there's a region here. You make lesions in many different ways. The whisking stops, but if we go to make lesions sort of in a shell surrounding what we claim is this pattern generator, which are these gray regions, then there's no effect on whisking. So we claim then, okay, that there's really this independent oscillator, and this region is necessary. Units in this region are necessary for rhythmic motion of the vibrissa. So now we have to look at connectivity. Okay, so again, there's a lot of ways to trace connections. There's modern ways with viruses or transsynaptic viruses. One of the powerful tools is to use this pseudorabies, which is a herpes simplex virus, which is a retrograde virus that goes transsynaptic. You inject it in the muscles, it'll work its way back. It's a great way to start. The problem is it has different tropisms for different synapses, so it gives you sort of a rough idea. So all the data I'm going to show you then is actually taken with very classic chemical organic tracers, which I would argue are somewhat cleaner still. So we get into the Botzinger complex, and we know we're here because we're recording. And then after we record it, we know where our electrode is. So this is breathing. This is the electrical recording. Then we can deposit a little bit of dye. So we deposit a dye, which is an anterograde transporter. So it's going up the axons, biatenylated dextranamine. So this is where the dye is injected. This is the breathing center. And now we're going to look in the location where the neurons are that drive the oscillator for whisking. And you can just see this nice axonal field, right, of axons going into this region. And just keep in mind, though, that these axons they're just sweeping across this whole region. So I'll get back to this in a moment. So this is a statement that there are wires, there are projections that go from the breathing CPG to the whisking CPG.
How far are these cells from the actual motor neurons? I mean, how many synapses the pre-Botzinger?
Okay, so I'll get there next. And then these neurons in the whisking center, right? We want to know if these are going directly into the motor neurons. Okay. So they are labeled by pseudorabies, but it took quite a while, right? Which we believe is due to tropism. Okay. In independent experiments, which I'll show you, which were originally done by Fan Wang, our colleague Fan Wang, using. Okay, so the trick is that she had an animal in which the motor neurons are making the rabies glycoprotein, and then she'll come in with a rabies virus which is incompetent to produce glycoprotein. It'll go into the motor neurons, pick up the glycoprotein, go one more step, and it'll go into this region. Okay, so we're pretty competent. What I'm just going to show here is that we're going to make an injection of a retrograde label. This is neurobiotin. Into the facial motor neurons, which directly drive the vibrissa, and we're staining them here with a acetylcholinesterase stain. And if we inject now into the facial motor nucleus, and they look in the vicinity, which from our other experiments, the region we believe to be the oscillator, we see these brightly labeled punched cell bodies, okay, because we're picking up in the terminals. And in fact, if we just count across many different fills, again, we're putting the label here. It's being back transported to cells in different brainstem nuclei. This is the region of the oscillator, and these are other areas these are one of the 20 areas that are projecting to the facial motor neurons. Okay, so just the small cells, the parvocellular neurons. So this says that the whisking CPG is sending fibers over to the facial motor nucleus. And again, we injected here, and a retrograde virus took us back to the CPG. So we put all this together, right? So we have lesions, we have activation, we have anatomy, we have recordings. We Xerox a page out of Paxinos, which is overpriced anyway, and we put all these recordings on top of each other. And you know, I say as good as you'll ever get in biology, they roughly sit on top of each other. And we claim then this is an area which is driving, in which there's a whisker CPG. So just to summarize, what we found so far is that in a previous work, much of it done by Jack Feldman, the initial work from Jack, identified this pre-Botzinger complex, which drives inspiration. Okay, this projects over to the parafacial respiratory group and the Botzinger complex, which drives expiration. Okay, so let's just go back here. The inspiration generator sends a reset pulse to the whisker oscillator, and this then goes to the motor neurons that drive protraction. Okay, so there's a part of the story which I didn't go through any of the details, but you got the gist. Okay, which it turns out that the exhalation generator, the Botzinger, also drives facial motor neurons that are driving the pad. So when the animal is rapidly whisking, these motor neurons are pulling the whisker forward, these motor neurons are pulling the whisker back, and you get these very large, fast amplitude whisks. Okay, so the summary is that breathing coordinates rhythmic whisking. Okay, but I mean, so far this is only of interest for whiskerologists, right? So how does this relate to the larger world? Okay, well, we had some clues because we saw these fibers coming out of the breathing center. They didn't just stop where the whiskers were; they just kept on going, and it suggested to us that in fact breathing may be interacting with many other breathing centers. So I'm going to conjecture a little bit, but also this is work in progress as a collaboration with our friend Fan, and so just as by way of background, that within the brainstem, there are different different motor nuclei. There's the facial motor nucleus that's driving the whiskers. There's a trigeminal motor 5, which is driving the jaw. There's a hyperglossal, this is driving the airway. And then we can look, and they're already known over time. People have found different oscillators. Oscillator for the tongues, the premotor nucleus, the oscillator I just described for the vibrissa. These two are known now to be giving input from the breathing center, the pre-Botzinger. This is known by anatomy. This is known by anatomy and physiology. So can we actually think of extending this, if there's a possibility that the breathing center is actually projecting to all these other motor nuclei? This is the motor nucleus driving chewing, the gigantocellular or orofacial branch. So this is the idea. We go in, we start with an animal which enables us to map projections from the muscle to the motor neurons, from the motor neurons to the premotor neurons. Okay, sort of a mixture of technology from Fan Wang's lab and Ed Calloway's lab, and I've already showed this for the vibrissa system. At the same time, we injected a dye with a different fluorescent protein that into the tongue, and it's going back to this premotor area for the hypoglossal nucleus. This is the oscillator for licking, okay? And then we're going to come in now with an antegrade transporter, which is Sindus virus, or which is biotinylated dextranamine, and look at the connectivity between these areas. And sort of the data so far, this is work in progress, suggests the following picture: that the pre-Botzinger cells, beside driving the phrenic motor neurons, just to drive breathing, okay, are also sending out these axons like a backplane in your computer. And the different premotor nuclei are just sitting there, okay? And so the facial motor neurons are picking up input. The breathing neurons are picking, swallowing neurons are picking up this input. Licking neurons are picking up this input. So we think that breathing center is sort of acting as this master clock for all these different behaviors to synchronize all these different behaviors. So that's, this is our ongoing idea. Now, this is what's happening at the brainstem. Now, I want to pick up a little bit of work that Dima he'll talk this afternoon, but I'm going to pick up a slide from his work. At least let's look at, licking, chewing have to do with taste. Whisking has to do with touch. Okay, breathing also has to do with smell. So let's see if there's any possibility for a common carrier between olfaction and touch. Okay, so I mentioned early on that the rats actually know where an object is because they can report to you whether they touch a pin at this position or this position. This is an experiment done by Summer Mehta in rats. There was very nice work by Dan O'Connor a couple of years after that in Karel Svoboda's lab, where they got this paradigm working with head-fixed mice. And one of the nice things about doing this in mice is now you start to pick up genetic control. But also what Dan showed is that if you remove temporarily sort of barocortex, primary vibrosensory cortex, this is done by putting mucimol, which turns on GABAergic inhibitory cells. You knock out vibrocortex, and the animals can no longer do this task. So anything that codes, okay, where the vibrissae are in space really requires primary vibrocortex. Okay, so let's then see what the nature of the signals are.
So this doesn't affect the movement. There's no movement here? It's an active touch, right?
Yeah, this is the politic thing to say is we sort of have a fight about this. Okay, so what's happening to the movements has not been well recorded. Okay, but the important point is they can no longer do this discrimination task.
Yeah,
Okay.
Okay.
Right, okay. Okay, so I'm going to record now in cortex, moving out of the brainstem, signals coming up from the follicle into the secondary neurons in the trigeminal complex, through thalamus, into cortex. And we see what the representation is. So the one question is that if we have a representation which is driven by this whisking clock, okay, then it's really just setting the phase, right? The amplitude information should be lost. The offset, the region of interest information should be lost. So while in the laboratory, right, we described vibrissae position in terms of angles, when we did this decomposition, right, just mathematical decomposition, we got it into sort of phase variables. And that's the variable that's being driven by the whisking clock. So again, the angle looks like there's these slowly varying parameters plus a rapidly varying phase. So the question is, does the animal actually code, okay, where contact occurred with respect to this phase variable? So this is the thesis work of the late John Curtis, and I'm just taking one picture to summarize that in fact vibrissae cortex codes vibrissae touch by phase in the whisk cycle. Now if the animals again are whisking rapidly, phase in the whisk cycle is the same as breathing, okay? And the experiment is the following. The animals are trained to whisk and to contact an object. If they contact the object, they get a reward. We know when they contact it because the object is a little piezo material, so you get a burst of change in electrical signal from the contact. And we're also monitoring this with a camera, etc. So we know where in the whisk cycle they made contact. The experiment is rigged that on different trials, the animals make contact at all possible phases, and at the same time, we're recording single units from primary sensory cortex. So this is one cell, an example of one cell, okay? And what's happening is that, in principle, it could have contacted at any place in its whisk cycle and gotten a barrage of spikes, and the spikes could have not depended where in the whisk cycle it is. But this is a cell that has a much stronger, 3 times higher firing rate if it touched here versus touched here, okay, in the whisk cycle. So this is tuned, okay, to phase in the whisk cycle. If we go back into absolute angle in the whisk cycle, there's absolutely no tuning. Okay, so the rats are coding their sensory information based on phase in the Wisp cycle. So there's a very nice experiment done in Dieter Rinberg's lab where they looked at the olfactory response in the olfactory bulb, recording from mitral cells. I don't want to take any part of Dima's thunder away, but it was a beautiful experiment. There's air that's flowing, and an odor is introduced into the airflow. Okay, and where you are in the breathing cycle was recorded very nicely by recording pressure in the nasal cavity. So you can see this is inspiration, this is the start of a cycle, and this is expiration. So if you just align the spikes based on the onset of the odor, okay, you can see something's happening from here to here, but the timing has tremendous jitter. If you now realign it based on relative to the onset of inspiration, which is here, so this is the breathing cycle, so this is 0, and this is 2 pi before you start another breathing cycle. So now it's rearranged with respect to phase in the breathing cycle. Different periods in time are now warped. You get this very, very sharp response. So this is the response in odor, okay, relative to the respiration cycle, which is equal to the Wisp cycle, okay? So the bottom line from this, we suggest, is that the rat is listening, right, to, in the case of smell, the rat just listens to her own breathing. Data from Ma's lab at Yale show that there are receptors in the olfactory bulb that not just respond to odors, but respond just to the flow of air. So they have a reafferent signal that tells them where they are in the breathing cycle. Data from some time ago in my lab from Michael Fee showed that in fact there are neurons in cortex, primary sensory cortex, that actually respond to where the whiskers are in space in the absence of contact. So somehow, olfactory information comes in at the rostral pole, vibrissae information comes in at the level of the brainstem, at the pons, and these things have a common carrier frequency. And it's just a conjecture that maybe nature is using this common carrier as a way to synchronize signals. So the lesson is that a common clock for phase sensitivity occurs in sniffing and whisking, and then we conjecture that inhalation may be a master clock that binds percepts based on other orofacial inputs like taste, and that should be that. Okay, thank you.
You're trying to say that the breathing is kind of a master clock. However, it's possible that it's a kind of a control loop that depends on behavioral targets. So in one case, breathing would master that clock, and in other case, another clock will drive another clock, depending on behavior.
What's the other clock?
Any of those clocks. There are clocks of breathing. There are clocks of whisking. There are or any other.
So there's many clocks. I mean, there's a looking clock. I didn't show the raw data. I mean, which we found. I'm sure I suspect there's a chewing clock, et cetera, et cetera. But the connections are not bidirectional. It's not a lattice of oscillators, each with bidirectional connections, and maybe depending on the activity, that one clock will lead in phase, right? That the phase relationship could change. I mean, it's the connections are unidirectional, and the thing you have to think about is, I mean, we came into this perhaps biased by Kuramoto and all these beautiful physical theories, but you cannot block the airway. Okay, you block your airway for one beat and you'll choke and you'll die. So it's not a good thing, and so you want a system that acts very quickly, and that's just worth thinking about why you can have a dominant clock. And there's sort of a hierarchy, right? Like swallowing could turn off breathing, but then breathing looks like it's projecting to everything else and dominating this hierarchy.
So there are no links, so there's no connections whatsoever from the other.
Back? No, there's no connection back. I mean, no in anatomy is a slightly dangerous term, but at some point you do enough tracing experiments, and you'll,
Yeah, the intriguing issue
going on with this question is that we can stop the breathing, for quite a while. It's not 1 second, it's much more, it's 30 seconds. It's a long time. So what you mean is that the breathing clock still works when I stop the breathing by other means and influences the rest, or something else? Because I do not understand how you can rely on a system that can be stopped for 30 seconds or more.
So we can voluntarily stop breathing. I don't know what happens to the Bottinger complex as we voluntarily stop breathing. Right? It's a point, okay? But if you stop breathing, you're also not sniffing.
Not sniffing, but I could move my hands and the brisa and face,
okay,
and
but
tongue and
that happens, and that happens. Okay.
So then the master clock does not work, and I do not rely on it.
Okay, so then
it's not the master clock.
So that's a good. That's an interesting experiment that has to be done. So suppose you believe that then chewing and licking, which are going to govern taste, could go on in the absence of breathing, and well, I mean, you can't.
Yeah, we can stop the clock. We can stop the breathing. I can show you how to do it.
Well, you did it.
You did it. Right? You're not stopping things. It's actually starting things, which is the hard part.
No, you can stop it for almost 2 seconds.
No, that's.
So maybe we'll discuss it after Dima's talk. I don't know. Will you talk about this, Dima?
A little bit.
Okay, so I mean, the deeper issue is that in the absence of the breathing clock, to the extent that it's, you do use this to synchronize other behaviors, are you actually affecting your percept if you stop breathing? So this is an experiment that has to be done. This has to be done.
This notion that location of object is encoded in the face of the Mishkenot. Is it the fact that in normal behavior rats don't move their heads when they're doing
Mishkenot? But they do. They head bob. And so that's another. There was a head bobbing CPG, which I didn't mention, but it's there. And so, I mean, this is sort of the program, right? To go into all the different muscle groups with these, rabies, viruses. The details don't matter, and see, to what extent we can push this program.
Is that locked to breathing too?
Yes, that's an easy experiment to do. Yeah, that's locked to breathing.
So all these things that activate facial muscles. So are these resets and things designed to prevent contrary muscle activations?
Yeah, that's the idea. I mean, that's the idea.
Does it work that way? You mentioned this one in the nose that moves the pads. Is it such that you don't get it the wrong way?
Well, so we never, I mean, again, you're picking up a little bit of a weak point. Like, we never do a surgery, right, to prevent this from happening. But that's sort of the idea. I mean, that these behaviors can run autonomously. To the extent that there's a bigger issue here, these behaviors run autonomously, licking, chewing, you do this all the time, but at times they conflict, right? So you have to establish a hierarchy. But we haven't done a critical test yet of that. But that would be, that's our grand hope, is that somehow this is maybe a model for neural processing in general that runs autonomously, but then all of a sudden things could conflict. Yeah. So it's on the agenda. You should come and do an experiment.
I know that Tom Sears used to sort of, go through this hierarchy of, breathing, speaking, singing, and then the highest level of control is saying absolutely nothing. But
well, you have a good voice.
But I just like, would you like to comment on that? I mean, here you've got a whole lot of very closely related behaviours, as you were just saying, but isn't there evidence that other forms of movement too are phased into the respiratory cycle? Is that?
Okay, so I mean, there are other things that happen. I mean, you run, right, and eventually block, but I think that just happens for other reasons because of compression of your chest. I'm supposed to say this. I mean, Noam has some beginning to have some data about things related to breathing. I mean, this is the only hard data I know. There's a lot of soft data out there. This is really the only hard data that I know of. And yeah, so I want to be careful. So we're sort of redoing it, but you know, and you give credit. I mean, it's sort of like Welker's paper. It's an anecdotal observation, but most of his I say all of his observations actually turned out to be true. So I suspect he did a lot of it that just said, oh, we did one. You know, it sounded cool.
If I may add, a soft fact is related to the dance thing we talked about yesterday. Playing violin, running, you must coordinate it with breathing. You must.
You
when I run, when I jog, I coordinate it with breathing.
But I mean, you do. But I mean, I think that the fun thing, this gets back to your previous question, is that your perception, for some reason, is also locked to this cycle. And I have to say, every time you breathe, right, you stretch the muscles on your face. So the rat, I mean, I didn't show the data here, but the rat knows when the rat's cortex knows when the rat breathes, not because of direct projections, right, not because of collaterals coming out of these brainstem oscillators, but just because of stretching of the face. All right, that's projected to cortex. Yeah.
Nikki or Chaim? I guess there is a conflict.
So I had one figure that I had to skip just for the time, but it was from another work by O'Connor that just was published last year, I think, or beginning of this year, where they did this illusory pole. So they can stimulate optogenetically these therefore neurons, and actually one of the figures shows that actually the timing related to the place of the whisker is not as tightly important.
Okay, so let me criticize that paper, since I spent 3 hours arguing in Bob's why they're wrong, but he works there, I don't. So let me explain the paradigm. In order for this paradigm, there's a couple of strategies the rat can use to tell where the pole is. So these are two extremes. One strategy, you're whisking back and forth all the time, and you touch. Okay, so in order to show that, then you have to take videographs and show the distribution of positions during the task. So when in Summers' thesis or paper, lost paper, the animals are spanning the entire space. In the work referring to from Carlsbad, which is Dan O'Connor and Andrew Hires, okay, the mouse takes the other sort of extreme. It puts its whiskers out, and it's hanging out in the position of one of the poles, which is the rewarded pole. Okay, then you hear the pole drop. So if it hits you, you know it was in the rewarded position. If it doesn't hit you, it's in the unrewarded position. So I say it's a different task, okay, because they're not sweeping their whiskers. And I didn't review the paper, but I am going to write a news and views, which will be published in a month, so we could. I don't take it lightly. Yeah.
I'm confused about the face code. Maybe for this task is enough, but in general, if the animal needs to know the position of an object,
yeah,
how do you go from face to position?
Okay, so you can. I don't know why the animal chose this. We could argue about, but in motor.
That's the question.
Okay, but the thing is, okay, what does it mean to know? So part of Dan Hill's thesis is reported from M1, from motor cortex. So these are regions that project down, okay, and not just activate the CPG, but activate these other areas that control the amplitude in ways that aren't well understood yet. So ongoing project. But neurons in primary motor cortex code the absolute angle or the amplitude. So that information exists.
It's a different code then. It's an independent code.
It's independent. Yeah, it's independent, but it's there, right? So then, the information is there in a place that the animal could veer in that direction and go off in that direction. Okay, so I didn't show that data. That was published about 2 years ago. You even argued with me about it, but maybe didn't know what it was for. It was arguing me about phenofactors.
So last question from
David, there's an actual example where breathing and let's say usage and detection with whiskers is decoupled, and that's right in front of your doorstep. the seals. They do phenomenal things in using their whiskers in detecting, but they stop breathing while they dive, obviously.
But they're not sweeping their whiskers.
They're not,
They're not
sweeping
sweeping.
their whiskers, but you made a point that all of this is connected to the detection, like in this experiment or so, where you sweep the whisker to detect. Obviously, you don't need the sweeping to detect, at least in some animals.
So there's no argument. The animal
So
is
is the argument then that you need to coordinate with the sweeping, with the breathing while you're sweeping, or while you're detecting? Do you depend on the breathing to detect?
So you don't depend on the breathing to detect. Okay. I guess so what I'm saying is there's a clock that sweeps, so you have to know you're getting a reafferent signal from that clock because that tells you where in the sweep you are.
Yeah.
So the difference in the seal is they're just detecting something. I mean, here you're detecting contact and you're asking at what position relative to the face did that contact occur. So that's the difference of the rat
But
versus
isn't
the
that sort
seal.
of turning things upside down? If you are sweeping, then you have a problem, and you better know what you're doing while you're sweeping to make a good detection.
No, I don't see. I think the problems break up because detection is just saying, did something hit the whisker? And then I'm saying, okay, something hit the whisker, and on top of that, I have to know the angle of the whisker.
Well, they do a whole lot of intricate stuff. I mean, following fish eddies or so on trivial feet, I guess.
So I can't really talk about the seal if I could. We don't really like the seal, to be honest. But I mean, you could take another strategy that you get a differential pressure on one side versus the other. You reorient your body until you get some equal pressure. Then all of a sudden you're tracking. So maybe that's a way to do it. They don't need to sweep. Okay. I just sort of given that the animals sweep, then there's sort of two. There's the detection task, and then there's the positional task as to where the object was. It's just like in vision. You want to know if photons came in, but you also want to know the orientation of the eye, so you know what the direction of the light was.
So in our function, mice really use the breathing clock to process this information. But humans can detect the odor in any phase of the sleeping cycle. The detection doesn't depend on the sleeping phase. You do long inhalation, and during the inhalation you pump an odor, you know the odor, you know the concentration. So it may be just kind of opportunistic use of this.
Okay, first I'm confused because the data in your own paper, which is
I
mine, but
agree.
not human, had preferred phases.
Yes, and the data is there, but it's hard to make a link. It's really hard to make a link to perception.
Okay, so it's a conjecture.
Yep.
And it's a fun talk.
Yeah.
And the data's the data, but it's interesting to think of maybe it's a perceptual clock. And there's something that has to be examined experimentally.
Okay, so I think we should all thank the speakers.